Hi, in this session we will be discussing about biological synthesis of silver nanoparticles. And biological synthesis or green synthesis is the synthesis of nanomaterial using biological organism, and we will be mainly discussing about synthesis of nanoparticle using plant extract, bacteria and fungi. So, nanotechnology is the concept of handling and usage of resources having a size of 1 to 100 nanometer. And those particles which are having a size to 1 to 100 nanometer are usually called as nanoparticles. And nanotechnology deals with handling and usage of these nanoparticles. And recently, scientists have started focusing more and more on nanoparticles due to their different magnetic, optoelectric, and physiochemical properties. And they have different properties or magnetic, optoelectric, and physiochemical properties due to variations in their size and shape distribution. So, nanoparticles have a variety of applications. That is, they can be used as catalysts, as chemical sensors, as electronic components, for imaging, for medical diagnosis and medical diagnostic protocols, as pharmaceutical products, for drug delivery, and they have this wide variety of applications due to their different properties or novel properties like physical, chemical and optoelectronic properties. And nanoparticles are categorized into various categories such as inorganic and organic nanoparticles. And under inorganic nanoparticles, there are semiconductor nanoparticles, metallic nanoparticles, and magnetic nanoparticles. And here we will be discussing about metallic nanoparticles, that is silver nanoparticles. So, silver nanoparticles. There has recently been a growing interest in gold and silver nanoparticles as they have superior characters with useful flexibility. And these silver nanoparticles has a substantial surface zone which gives it many important biochemical reactivity, catalytic activity, atomic behavior compared with bigger particles having same chemical composition. So these silver nanoparticles, due to their surface zone, they have superior qualities compared to bigger particles having same chemical composition. And the silver particles have received significant interest because they can be used as catalysts, plasmonics, in optoelectronics, as biological sensors. They have antimicrobial activities. They can be used for DNA sequencing and surface enhanced Raman scattering. They can be also utilized in climate change and contamination control, for clean water technology, energy generation, information storage, and biomedical application. These are the various applications in which silver nanoparticles can be utilized. And as already discussed, nanoparticles can be synthesized in two ways, that is top down approach and bottom up approach. In top down approach, we are converting a bigger molecule into smaller units and then converting into smaller nano-sized particles. And in bottom up approach, we are letting small molecules to assemble themselves to form nanoparticle. And green synthesis and chemical methods are different methods of bottom up approach. And under that, in green synthesis is the synthesis of nanoparticles using bacteria, fungi, plant extract, enzymes or microorganism. And the advantage of green synthesis compared to chemical and physical methods is that they are non-toxic, cost effective, and also environmental friendly, and they produce lesser toxic materials compared to other methods. So in bottom-up approach, nanoparticles can be synthesized using chemical and biological method, and in bottom-up approach, self-assembly phenomena of atom is utilized. That is, these atoms self-assemble themselves to form new nucleus which grow into particles of nano scale. And the reason that led to biosynthesis of nanoparticle is because it does not use any toxic chemical, and therefore it is a more environmental friendly process for synthesizing of nanoparticles. So development of green synthesis of silver nanoparticles is advancing as a key branch of nanotechnology, and in green synthesis we are using biological entities like plants, plant extract, plant biomass, microorganism for generation of nanoparticles, and this act as an alternative way against chemical and physical methods in an eco-friendly way. And the advancement or recent interest in green synthesis over chemical and physical methods are due to the fact that green synthesis is more environmental friendly, it is more cost effective, and it can easily be scaled up for vast scale synthesis of nanoparticles. And in green synthesis we are not utilizing high temperature, energy, pressure or harmful chemicals are not used. And green synthesis, according to the biological material we are using, can be categorized into three. In first one, we are utilizing microorganisms like fungi, yeast, bacteria and actinomyces. Second category is green synthesizing using plant and plant extract, and third category is green synthesizing using templates like membrane, viruses, DNA and diatoms. These are the three categories of green synthesis. So first we are going to discuss about synthesis of silver nanoparticle using bacteria. So this is a schematic diagram for synthesizing of silver nanoparticle using bacteria. To bacterial biomass we are adding AgNO3 solution, and this solution by the activity of enzymes in the bacteria will be converted into nanoparticles, and characterization of these nanoparticles can be done by using different tools, and after characterization it can be used for various applications like antibacterial activity, air disinfection and food packing. So inorganic materials are produced by bacteria. Usually these inorganic materials are produced either intracellularly or extracellularly. So they make a potential biofactories for synthesizing nanoparticles like gold and silver. And silver nanoparticles are not to be biocompatible with these bacteria, but certain bacteria are also resistant against silver nanoparticles. Therefore these bacteria aggregate the silver on the cell wall, and they can be utilized in industrial recovery of silver from ore material. And different bacteria are utilized for synthesizing of silver nanoparticle. An example of such a bacteria is Pseudomonas schutzeri, and it is used for synthesizing of silver nanoparticles. And using this bacteria, we can synthesize nanoparticles up to 200 nanometer size. Another one bacteria which we are using is psychrophilic bacteria, can also be utilized for synthesizing of silver nanoparticles. Then Bacillus licheniformis is used for synthesizing of silver nanoparticles, in which we are adding this Bacillus licheniformis biomass onto aqueous AgNO3 solution, and after addition of this bacteria, this solution changes from whitish yellow to brown color, and this change in color indicates the formation of silver nanoparticles, and usually nanoparticles of 50 nanometer size can be synthesized using this bacteria. Another example is use of Staphylococcus aureus, and it can be used for synthesis of nanoparticle. And for quick synthesis, enterobacteria can also be utilized. Another bacteria is Proteus mirabilis. This can be used for synthesizing silver nanoparticle, and usually extracellular and intracellular synthesis can be done using different kinds of broth and incubation of bacteria. And using bacteria for green synthesis is more flexible, it is reasonable, and it is also suitable for large scale synthesis of nanoparticles. And Klebsiella pneumoniae can also be used for synthesizing silver nanoparticles from silver nitrate. And other bacteria which can be utilized for synthesizing silver nanoparticles are Enterobacteria cloacae, Escherichia coli, and Klebsiella pneumonia. And usually using these Klebsiella pneumonia, the synthesis of silver nanoparticle is very fast, and within 5 minutes silver ions can be converted into nanoparticles by these bacteria. And there are many advantages of using bacteria for synthesis, but there are certain disadvantages also for using bacteria for synthesizing of nanoparticle. The disadvantages are that the rate of synthesis is very slow, and only a limited number of size and shapes can be obtained by using bacteria as nanofactories. So therefore fungus and other plant materials are utilized for synthesizing of nanoparticles. Next is synthesizing of silver nanoparticle using fungi. In that we are adding to a biomass of fungi, we are adding AgNO3 solution, and here synthesis occur both extracellularly and intracellularly. After synthesizing, we are characterizing the nanoparticles, and they can be utilized for different applications. So fungus have great potential for synthesizing of metallic nanoparticles due to different properties of fungus, and those properties which make them suitable as nanofactories are that they have metal bioaccumulation capacity, they have tolerance, they have high binding capacity, and both extracellular and intracellular uptake take place using fungus, and also it's comparatively easier to handle compared to bacteria. And fungus can be used through different methods for synthesizing of nanoparticles, and fungi secrete enormous enzymes, large quantities of enzymes, which can be utilized for reducing the silver nitrate solution into silver nanoparticles. And the mechanism of nanoparticle synthesis in fungus based system usually The nanoparticles may be formed on the surface of mycelia, or in certain cases they can also be formed on the solution. And when they are formed on the surface of mycelia, usually first the silver nanoparticles are absorbed on the surface of the fungal cells due to electrostatic interaction between the cells and the particles. And then these particles are reduced by the enzymes present in the cell wall, which lead to formation of Ag nuclei, and from it nanoparticles are formed. And the shifting from bacteria to fungus to produce nanofactories often more advantages. That is, it has a straightforward downstreaming process, then higher amount of proteins are present, and therefore we can drastically increase the profitability of synthesis using fungi. And extracellularly also nanoparticles can be produced using fungi. And extracellular synthesis is useful, and during extracellular synthesis the nanoparticles will not bind to the biomass. They are actually produced in the solution, and this method is more eco-friendly and more straightforward, and this lead to utilization of fungus for synthesizing of nanoparticle. And the reaction rate is also an important supporting factor for utilizing fungus for synthesis of nanoparticles. Next is different fungi which we can use for extracellular synthesis. So Fusarium oxysporum can be used for synthesizing silver nanoparticle, and it has antibacterial effect. Then Aspergillus flavus can be utilized, and using Aspergillus flavus we can synthesize nanoparticles of 8 to nanometer range. And Cladosporium cladosporioides is also utilizing for synthesizing silver nanoparticle extracellularly, and also using Penicillium cellutanum, which is a fungus which has been isolated from coastal mangrove sediments. Also silver nanoparticles can be synthesized using silver nitrate solution. Then Fusarium oxysporum can synthesize nanoparticles of 5 to 15 nanometer size. Then extracellularly nanoparticles can be synthesized using Aspergillus fumigatus, and Aspergillus tertius can synthesize spherical nanoparticles of 1 to 20 nanometer size. Then Trichoderma ricci can synthesize nanoparticles of 5 to 50 nanometer, and Verticillium can synthesize monodispersed silver nanoparticles. And usually in using Verticillium, it is this fungal biomass is exposed to aqueous solution of AgNO3, and thereby spherical shaped nanoparticles of 25 plus or minus 12 nanometers are synthesized, and they are usually synthesized below the surface of the fungus cells, and they have different shapes like spherical, triangular or hexagonal shape. Next is Aspergillus flavus also can be used for synthesizing nanoparticles. So fungus is more suitable material for synthesizing nanoparticles compared to bacteria, because large scale synthesis can take place using fungus, and also extracellular synthesis of nanoparticles can take place using fungus. Next is synthesis of silver nanoparticle using plant and plant extract. Here plant extract is added to silver nitrate solution and nanoparticle is synthesized and characterization and different applications are done. And metallic nanoparticles using plant extract was synthesized in 2013 for the first time by Gardia Torsday. So in 2013, for the first time, metallic nanoparticles were synthesized using plants, and Gardias did experiment using alpha sprouts, and it's the first synthesis of silver nanoparticle using living plant system. And in this, alfalfa roots absorb the silver from the medium and they are transported into the shoot, and in these shoots the silver atoms aggregate themselves to form silver nanoparticles. And it is demonstrated that using plant extract for synthesizing of metal nanoparticles using metal salt solution is advantageous because it can be done within a short time period at normal room temperature. And after selecting the plant for preparing the plant extract, the different parameters which affects the synthesis of nanoparticles are the concentration of the extract, temperature, metal salt, pH and contact time. And in addition to that, the plant species which we are selecting also has an important role. And the benefit of using plants for synthesizing nanoparticles is that they are easily accessible, it is safe to handle, and also they have a large range of active agents that helps in reduction of silver ions into nanoparticles. And mainly different parts of plants can be utilized for synthesizing silver nanoparticles, that is roots can be used, latex, stem, seed and leaves can be used. And an interesting fact about utilizing plants for synthesis of nanoparticle is that as such these plants has many active agents which make them suitable for stabilization and reduction of the silver ions, and thereby we can produce nanoparticles of stable and shape controlled nanoparticles. And the different bioactive agents which are present within the plants which help in reduction and capping of nanoparticles are biomolecules like terpenoids, polysaccharides, phenols, alkaloids, flavones, amino acids, enzymes and proteins, and also pigments like chlorophyll pigment, then methyl chavicol, linalool, caffeine, eugenol, ascorbic acid, theophylline and other vitamins are also act as reducing agents within plants. And green synthesizing using plants is more faster compared to microorganisms such as bacteria and fungi, and also there are many advantages like rapid growth and a single step technique, then economical protocol, they are non-pathogenic and they are also eco-friendly for nanoparticle synthesis. And few examples of synthesizing silver nanoparticles using different plant extract are Ananas comosus has been utilizing for synthesis of silver nanoparticles, and the synthesized particles were characterized using different tools, and nanoparticles of size 12 nanometer was obtained. Then by using Argemone mexicana leaf extract, nanoparticles of 13 nanometer size were obtained. Then by using neem and trifla, using aqueous silver nitrate solution, nanoparticles of size range 43 to 59 nanometer was obtained. And using peanut shell extract, nanoparticles with diameter 10 to 15 nanometer was obtained, and these nanoparticles had antifungal activity. And then using Malus domestica, nanoparticles of 20 nanometer was obtained. So these are the different methods for synthesizing of nanoparticles. Biosynthesizing of silver nanoparticles. Both bacteria, fungus and plant can be used, and each has their own advantages compared to physical and chemical method. And using plant is more advantageous than using microorganism and like bacteria or fungi, because they are non-pathogenic, and also rapid synthesis of nanomaterial can take place. Next, we are going to see some of the applications of silver nanoparticles. They can be divided into different categories like medical application, environmental application, consumer goods and personal care products, food, electronic component and transportation, antibacterial activity, antimicrobial activity, antiviral activity and antifungal activity. So nanoparticles synthesized using silver had antibacterial, antimicrobial, antiviral and antifungus activity, and they can be used for synthesizing of biosensors for food analysis, then sensors, electrodes and integrated circuits. In environmental application, they can be used for water purification, then biological wastewater and groundwater disinfectant, surface disinfectant, wastewater treatment. And in medical application, they can be used as dental cements, healing of burns and wounds, skin therapy, reconstructive surgeries, medical devices, targeted drug delivery, cancer therapy. And as consumer goods and personal care products, they can be used in textiles, paints and cosmetics, toys, filters, kitchen utensils, toothpaste and different cosmetic products like shampoos, deodorant, etc. And they can be also used for food safety and food packaging. So these are the various applications of silver nanoparticles. Thank you.